Have you ever wondered how scientists can map the tiny, complex structures inside your brain? Today, we're diving into a groundbreaking study that's revolutionizing how we visualize the thalamus, the brain's sensory relay station. This region, packed with dozens of minuscule nuclei, has long been a challenge to study due to its intricate architecture. But researchers have cracked the code, and the key lies in a surprising imaging technique. Let's start with the basics. The thalamus acts like a neural traffic controller, routing sensory and motor signals between the brainstem and cortex. But its nuclei, each with distinct functions, are so small and densely packed that traditional MRI scans struggle to differentiate them. Think of it like trying to spot individual trees in a dense forest using a blurry camera. Standard T1 weighted images, while common, lack the contrast needed to resolve these tiny structures. So how did researchers solve this? They tested multiple imaging methods, MPRAGE, FGATIR, and advanced quantitative maps like T1 relaxation time measurements. But here's the twist: they didn't just rely on intuition. Instead, they used a 3D UNet neural network, a deep learning model designed for medical image segmentation, to analyze which technique provided the clearest results. To refine their findings, they employed a technique called gradient-based analysis with Monte Carlo Dropout, which essentially asks the AI, which parts of this image are most critical for accurate segmentation? By running this test thousands of times, they quantified the importance of each imaging contrast. The results: T1 maps, images that measure how quickly brain tissues return to equilibrium after a magnetic pulse, emerged as the gold standard. Why? These maps highlight subtle differences between nuclei that other methods miss. For example, while multi-T images, which capture multiple time points, initially seemed promising, the study found that T1 maps alone captured nearly 90% of the critical contrast information. Even PD maps, proton density, added minimal value beyond T1. Essentially, the key data in multi-T scans is already embedded in T1 maps, simplifying workflows and reducing processing needs. But here's the catch: T1 maps aren't perfect. While they're efficient for AI models, multi-T images offer richer visual contrast that human experts might prefer for manual analysis. It's like comparing a high-resolution photo to a simplified sketch. Both have their place. The study's real breakthrough was developing an overall importance score, OIS, a metric that quantifies how much each imaging modality contributes to segmentation accuracy. This tool could redefine how researchers evaluate imaging techniques in the future. What does this mean for neuroscience? Better thalamus segmentation could unlock new insights into diseases like Alzheimer's and Parkinson's, where thalamic dysfunction plays a role. It also improves precision for treatments like deep brain stimulation, where targeting specific nuclei is critical. And for AI researchers, this work highlights how combining deep learning with traditional imaging can push the boundaries of what's possible. So the next time you hear about brain imaging advances, remember, sometimes the most powerful tools are the ones we've already mastered. Thanks for watching, stay curious, and if you want more deep dives into neuroscience and AI, hit subscribe.